Hello everyone. Welcome to the webinar session on the foodborne disease part 1. I am Kavita Kulkarni, Scientific Affairs and Education Manager, Food Safety Division, 3M India Limited. I thank AOAC International India section for giving me this opportunity to present this important session to you all. Let's start with the session. In this session, I will be discussing about the pathogens causing foodborne diseases. I'll be taking you through the bacterial agents, pathogenesis, clinical features, isolation and identification, association with foods, their mode of action for the following organisms, which are Aeromonas, Hydrophila, Bacillus ceres, Brucella, Campylobacter, Clostridium botulinum, Clostridium perfringens, Chronobacter sakazaki, and Escherichia coli. Now coming to the Aeromonas hydrophila. Aeromonas hydrophila bacteria are gram-negative straight rods with rounded ends, bacilli to coccobacilli shape, and are motile by a polar flagellum. They can grow at temperatures as low as 4 degrees centigrade. They are facultative anaerobes which cause opportunistic infections as well as occasional case of food and waterborne illness. They are present in fresh and brackish waters. Aeromonas hydrophila is an opportunistic pathogen in humans, meaning they rarely infect healthy individuals, causing extraintestinal and intestinal infection. It is an animal pathogen leading to infections in fish, amphibians, and mammals. They undoubtedly form biotic biofilms. Now coming to the pathogenesis and clinical features. Because of its structure, it is very toxic to many organisms. When it enters the body of its victim, it travels through the bloodstream to the first available organ. It produces aerolysin, cytotoxic enterotoxin that can cause tissue damage. A. hydrophila is widely considered a major fish and amphibian pathogen, and its pathogenicity in humans has been recognized for decades. Now coming to the pathogenesis and clinical features, wherein we will be discussing about the animal pathogen leading to infections in fish, amphibians and mammals, and also we will be discussing about the human diseases caused by Aeromonas hydrophila. So under animal pathogen, if you see here, the disease might be precipitated by low oxygen concentrations, nitrite-induced methemoglobinemia, rapid temperature changes, stress associated with spawning, decreased immunity, and physical trauma. When infected with A. hydrophila, fish develop ulcers, tail rot, fin rot, and hemorrhagic septicemia. Hemorrhagic septicemia causes lesions that lead to scale shedding, hemorrhages in the gills and anal area, ulcers, exophthalmia, and abdominal swelling. Coming to the human diseases, it causes gastroenteritis, mostly in the people who have compromised immunity. This bacterium is linked to two types. The first type is called as rice water diarrhea, and the second type is the dysenteric gastroenteritis. Dysenteric gastroenteritis is the most severe out of the two types, which can last for several weeks. Association with foods, their mode of action. This organism is found in many foods, especially foods of animal origin. It may be found in milk, finfish, seafood, red meat, and poultry. In some foods, it occurs at a level of 10 to the power 5 cells per gram or per ml. The strains have been implicated in the spoilage of foods. Because of the psychotrophic nature, they can grow in foods at refrigerated temperature and even from a low initial load, which can reach a high population with time during storage. Their presence in high numbers in food can be controlled by heat treatment, preventing post-heat contamination, and using one or more of the growth limiting parameters such as low pH and low water activity. Now coming to the isolation and identification. Protease peptone and beef extract provide essential nitrogenous compounds. D-xylose is source of carbon and energy for these organisms. Aeromonas species do not ferment xylose, and oxidase test can be performed on colonies that do not produce acid. The current formulation of Aeromonas selective agar, which is BSIBG, the full form of that is bile salt irrigation brilliant green agar, is recommended for the isolation of Aeromonas species from food. It is oxidase negative and indole positive. Moving on to Bacillus series. Bacillus series is a gram-positive, rod-shaped, facultatively anaerobic, motile, beta-hemolytic, spore-forming bacterium commonly found in soil and food. Some strains are harmful to humans and cause foodborne illness, while other strains can be beneficial as probiotics for animals. B series are facultative anaerobes and, like other members of the genus Bacillus, can produce protective endospores. Here, if you see in this picture, you can see the spores also in the slide. So these are basically the endospores. Its virulence factors include cereolysine and phospholipase C. So Bacillus species, under Bacillus series group, it comprises 7 closely related species, which are B series, B anthracis, B thuringiensis, B. mycoides, and so on. The strains produce at least two types of toxins, emetic and enteric of enterotoxins, each probably associated with specific types of symptoms. Bacillus foodborne illnesses occur due to survival of the bacterial endospores when infected food is not or inadequately cooked. The two types of enterotoxins produce two types of symptoms. The enterotoxin associated with the diarrheal form is a heat labile protein, and that associated with the emetic form is a heat stable protein. Symptoms include abdominal pain, profuse watery diarrhea, and perhaps nausea, but no vomiting or fever. In the emetic form, symptoms are nausea and vomiting, followed by abdominal pain and diarrhea. In some cases, the symptoms are similar to those of staphylococcal gastroenteritis. Moving on to the isolation and identification. For the isolation and enumeration, there are two standards which are ISO 79331 and ISO 21871. Selective media for its isolation and identification, such as mannitol egg yolk polymyxin, which is called in short called as MYP agar, and polymyxin pyruvate egg yolk mannitol bromothymol blue agar, which is PMBA agar. Bacillus cereus colonies on MYP have a violet-red background and surrounded by a zone of egg yolk precipitate. Below is a list of differential techniques and results that can help to identify Bacillus cereus from other bacteria and other Bacillus species. Anaerobic growth, it is positive. Vogt-Prosker test is positive. Starch hydrolysis, nitrate reduction, and citrate utilization are positive. And growth at greater than 50 degrees centigrade is negative. Association with foods, their mode of action. In the diarrheal outbreaks, a variety of foods, including vegetables, salads, especially the cold salads, meats, puddings, casseroles, sauces and soups, have been implicated, mostly because of their improper cooling or storage conditions. However, in the emetic form, outbreaks mostly involve rice and sometimes other starchy food. The most predominant was fried rice in Chinese restaurants, which is also called as Chinese rice syndrome. If you see the slide over here, the rice, if you can just imagine improperly stored cooked rice. So which is the following of the boiling of preparation, the rice is kept at room temperature for a long time. So in such case, the surviving or the contaminated spores germinate and the cells multiply to high levels before the rice is served. So hence, the rice is one of the good carrier for Bacillus cereus. Brucella. Brucella is a genus of gram-negative bacteria named after David Bruce. They are small, non-encapsulated, non-motile, facultatively intracellular cocobacilli. They are gram-negative, non-spore forming, aerobic, small rods, pathogenic to animals and humans. They survive extremes in temperatures, pH and humidity, and in frozen and aborted materials. They infect many species but with specificity. They usually have two chromosomes and their replication and segregation are temporally organized. Brucella causes a zoonotic infection called brucellosis. The bacteria are transmitted from animals to humans by ingestion through infected food products, direct contact with an infected animal, or inhalation of aerosols. The disease is an old one that has been known by various names, including Mediterranean fever, Malta fever, gastric remittent fever, and undulant fever. Brucella organisms localize in the reproductive organs of host animals, causing abortions and sterility. Human brucellosis is considered as a life-threatening, debilitating disease characterized by weakness, fever, malaise, arthritis, osteomyelitis, endocarditis, or meningoencephalitis. Isolation. Growth seen on blood agar and chocolate agar. They are non-hemolytic, non-pigmented, small grey colonies. They might require 48 to 72 hours for discrete colonies to become evident. The carbon dioxide availability enhances growth. They are slow growers with fine colonies with punctuate colonies in 48 hours. Clear or white colonies on McConkey agar at 48 hours. When you do a gram staining, they are tiny, often faintly stained, gram-negative coccobacilli, but more often they will be mistaken as cocci. May retain crystal violet stain in blood culture smears. The biochemical test reactions are urea test is positive, oxidase test is positive, and they are non-motile. Remember this, look at this small box over here, which is yellow background. Brucellosis is a commonly acquired laboratory infection. So all laboratory microbiologists, be careful while working with this Brucella organism. All work on suspect Brucella cultures should be performed at a minimum of biosafety level 2 conditions with biosafety level 3 practices. Coming to the association with the foods and their mode of action. In infected animals, the organisms are located in the uterus of pregnant animals and in the mammary glands of lactating females. Thus, the pathogens can be excreted in milk. People working with animals can become infected with Brucella species. Also, people working with meat can also be infected. Consumption of raw milk and some products made from raw milk, such as some imported cheese, have been implicated in foodborne brucellosis. The cells survive for a long time in milk and milk products. Pasteurization of milk and milk products kills the Brucella cells. Control measures for foodborne brucellosis include pasteurization of milk, manufacturing of dairy products from pasteurized milk, and proper sanitation to prevent recontamination of pasteurized products. Campylobacter. Under Campylobacter, the two species which are more important or very important are these two foodborne pathogens, which are Campylobacter jejuni and Campylobacter coli. They are small microaerophilic, helical, motile cells found in the intestinal tract of humans, animals, and birds. Campylobacter species generally appear as curved or comma-shaped rods, and they are able to move via unipolar or bipolar flagella. They generally survive in environments with low oxygen. When exposed to atmospheric oxygen, C. jejuni is able to change into a coccal form. They are positive by the oxidase test and catalase test. They are non-fermentive and are best cultured at 42 degrees centigrade. Survival at room temperature is poor, but they can survive for a short time at refrigeration temperatures up to 15 times longer at 2 degrees centigrade than at 20 degrees centigrade. The bacterium dies out slowly at freezing temperatures and is heat sensitive. The cells are destroyed at temperatures above 48 degrees centigrade. Bacillus are gram-negative, curved or spiral. They are very motile, oxidase positive, catalase positive, and microaerophilic. Coming to the pathogenesis and clinical features. Campylobacter jejuni has a thermolabile enterotoxin that is responsible for enteric disease symptoms. The main symptoms are enteric and include abdominal cramps, profuse diarrhea, nausea, and vomiting. Other symptoms include fever, headache, and chills. An individual can have a relapse of symptoms after a short interval. Campylobacter can cause a gastrointestinal infection called campylobacteriosis. This is characterized by an inflammatory, sometimes bloody diarrhea or dysentery syndrome, mostly including cramps, fever, and pain. Most strains of C. jejuni produce cytolethal distending toxin, which inhibits cell division and impedes activation of the immune system. This helps the bacteria to evade the immune system and survive for a limited time inside intestinal cells. The organism produces diffuse bloody edematous and exudative enteritis. In some cases, a Campylobacter infection can be the underlying cause of Guillain-Barré syndrome. Revised ISO 10272 Part 1 for detection of Campylobacter species was published in June 2017, and Bolton broth and Preston are described for the pre-enrichment purpose, and using charcoal cephaloperazone deoxycholate modified agar base for isolation was recommended. So we learned in the previous slide also that biochemical test Campylobacter responds as oxidase positive and catalase positive. There are advanced methods which are PCR and LAMP techniques which can give results which are reliable and faster. Association with foods and mode of action. The foods can be contaminated directly with fecal material from animals and infected humans, or indirectly from sewage and contaminated water. They have been isolated at a very high frequency from raw meats, beef, such as beef, lamb, pork, chicken, and turkey, milk, eggs, vegetables, mushrooms, and clams. In heat processed food, their presence have been related to cross-contamination following heat treatment or improper heating. Clostridium botulinum. This is a gram-positive, rod-shaped, anaerobic, spore-forming, motile bacterium with the ability to produce the neurotoxin botulinum. This is widely distributed in nature and is assumed to be present on all food surfaces. In spore form, it is the most heat-resistant pathogen that can survive in low-acid foods and grow to produce toxin. The toxin attacks the nervous system and will kill an adult at a dose of around 75 nanogram. Growth of the bacteria can be prevented by high acidity, high ratio of dissolved sugar, high levels of oxygen, very low levels of moisture, or storage at temperatures below 3 degrees centigrade for type A. Coming to the pathogenesis. The botulinum toxin can cause a severe flaccid paralytic disease in humans and other animals and is the most potent toxin known to mankind, natural or synthetic, with a lethal dose of 1.3 to 2.1 nanogram per kg in humans. Clostridium botulinum is a diverse group of pathogenic bacteria initially grouped together by their ability to produce botulinum toxin and now known as 4 distinct groups, which are Clostridium botulinum groups 1 to 4. C. botulinum is responsible for foodborne botulism, ingestion of preformed toxinitis. Infant botulism is by the intestinal infection with toxin-forming C. botulinum, and wound botulism, which is the infection of a wound with Clostridium botulinum. Clostridium botulinum is commonly associated with bulging canned food. Bulging, misshapen cans are due to an internal increase in pressure caused by gas produced by the bacteria. Botulism poisoning can occur due to preserved or home-canned low-acid food that was not processed using correct preservation times or pressure. Foodborne botulism. Signs and symptoms of foodborne botulism typically begin between 18 to 36 hours after the toxin gets into your body, but can range from a few hours to several days depending on the amount of toxin ingested. So the following are the various symptoms which one encounters: double vision, blurred vision, drooping eyelid, nausea, vomiting and abdominal cramps, slurred speech, trouble breathing, difficulty in swallowing, dry mouth, muscle weakness, constipation, reduced or absent deep tendon reactions such as in the knee. Let's talk about the clinical features. Infant botulism and adult intestinal toxemia. We will be talking about these two features. Under infant botulism, if the disease is related to food such as honey, problems generally begin within 18 to 36 hours after the toxin enters the baby's body. Signs and symptoms include constipation, often this is the first sign, floppy movements due to muscle weakness and trouble controlling the head, weak cry, irritability, drooling, drooping eyelids, tiredness, difficulty in sucking or feeding, and paralysis. Under adult intestinal toxemia, a very rare form of botulism that occurs by the same route as infant botulism but is not that common in the adults, occurs rarely and sporadically. Signs and symptoms include abdominal pain, blurred vision, diarrhea, dysentery, imbalance, weakness in arms and hand area. Let's talk about the isolation and identification. In the laboratory, Clostridium botulinum is usually isolated in tryptose sulfite cycloserine, which is TSC growth medium in an anaerobic environment with less than 2% oxygen. So here is a jar, anaerobic jar for you all to have a look. Anaerobic environment can be achieved by several commercial anaerobic chambers that use a chemical reaction to replace oxygen with carbon dioxide. C. botulinum is a lipase-positive microorganism that grows between pH of 4.8 and 7 and cannot use lactose as a primary carbon source, with the characteristic which is important for biochemical identification of Clostridium botulinum. Association with foods, their mode of action. In a low acid canned vegetable such as green peas or green beans that are not heated enough to kill the spores, that is, a pressurized environment may provide an oxygen-free medium for the spores to grow and produce the toxin. However, pickles are sufficiently acidic to prevent growth. Even if the spores are present, they pose no danger to the consumer. Honey, corn syrup, and other sweeteners may contain spores, but the spores cannot grow in a high concentrated sugar solution. However, when this sweetener is diluted in the low oxygen, low acid digestive system of an infant, the spores can grow and produce toxin. But as soon as infants begin eating solid food, the digestive juices become too acidic for the bacterium to grow. The control of foodborne botulism caused by Clostridium botulinum is based almost entirely on thermal destruction of the spores or inhibiting spore germination into the bacteria and allowing cells to grow and produce toxins in foods. Conditions conducive of growth are dependent on various environmental factors. Moving on to the Clostridium perfringens. This is a spore-forming gram-positive bacterium that is found in many environmental sources as well as in the intestines of humans and animals. This is commonly found on raw meat and poultry. It prefers to grow in conditions with very little or no oxygen and under ideal conditions can multiply very rapidly. Some strains of Clostridium perfringens produce a toxin in the intestine that causes illness. Pathogenesis and clinical features. Everyone is susceptible to food poisoning from Clostridium perfringens, but the very young and elderly are most at the risk of Clostridium perfringens infection and can experience more severe symptoms that may last for 1 to 2 weeks. Complications including dehydration may occur in several cases. This organism can survive high temperatures. During cooling and holding of food at temperatures from 12 to 60 degrees centigrade, the bacteria grows. It can grow very rapidly between 43 degrees to 47 degrees centigrade. If the food is served without reheating or without adequate heating to which can kill the bacteria, the live bacteria may be eaten. The bacteria produce a toxin inside the intestine that can cause illness. People infected with Clostridium perfringens develop diarrhea and abdominal cramps within 6 to 24 hours. The illness usually begins suddenly and lasts for less than 24 hours. People infected with this organism usually do not have fever or vomiting, and the illness is not passed from one person to another. Let's talk about the isolation and identification. First step is the isolation. The selected followed by the presumptive confirmation. The selective colonies of Clostridium perfringens are selected from TSC egg yolk agar plates and inoculated into freshly de-aerated thioglycolate broth, which is followed by the biochemical confirmation. Use of lactose gelatin media for growth of Clostridium perfringens, and production of gas and color change from red to yellow, which indicates acid production for the positive tubes. Food poisoning caused by Clostridium perfringens may occur when foods such as meat or poultry are cooked and held without maintaining adequate heating or refrigeration before serving. The presence of small numbers of Clostridium perfringens is not uncommon in raw meats, poultry, dehydrated soups and sauces, raw vegetables and spices. The enterotoxin can be detected in sporulating cultures. The cells lose their viability when foods are frozen or held under prolonged refrigeration unless special precautions are taken. Such losses may make it difficult to establish Clostridium perfringens as the specific cause of a food poisoning outbreak. Next organism which we are going to discuss is Enterobacter sakazaki, or the new name is Chronobacter sakazaki. This was Chronobacter, which used to be called earlier as Enterobacter sakazaki, is a germ that can live in very dry places. This is found in dry foods like powdered infant formula, powdered milk, herbal teas, and starches. It has also been found in sewer waters. Getting sick with Chronobacter does not happen often. Chronobacter infections are often very serious for babies, though they can even die. Chronobacter infections can also be very serious for older people and people whose bodies have trouble fighting germs, like people with HIV, organ transplants, or cancer. Now let us understand the pathogenesis and clinical features. In mammalian tissue culture, the organism can attach to intestinal cells and survive internally in macrophages. This organism can attach to plastics and silicone rubber surfaces and grow in as biofilm. Enteral feeding tubes and feeding bottle teats can harbor the bacterium in larger numbers. Sickness from Chronobacter look different depending upon the person. In babies, especially less than 2 months old, these organisms get in the blood or make the lining of the brain and spine swell, which is causing leading to meningitis. Sickness from Chronobacter in babies will usually start with a fever and poor feeding, crying or very low energy. Some babies may also have seizures. Babies with meningitis may develop serious, long-lasting problems in their brains. For the rest of the people of all ages, this organism can cause problems in cuts, scrapes, or places where people have had operations or surgeries. Chronobacter can also get into the urinary tract. Older people and people with weakened immune systems are more susceptible for this illness. As per ISO 22964, the isolation step includes the Chronobacter selective broth and chromogenic Chronobacter isolation, agar. Enterobacter species are biochemically similar to Klebsiella, but unlike Klebsiella, Enterobacter is ornithine positive. So as we already talked about this, powdered infant formula is the most important food product which is associated with this organism. So powdered infant formula is not germ-free. Formula manufacturers report that it is not possible to remove all germs from powdered infant formula. At the factory, Chronobacter could get into formula powder from ingredients used to make the formula, or if the formula powder touches a Chronobacter covered surface in the factory. At home, Chronobacter could get into the formula if formula lids or scoops are placed on chronobacter covered surfaces and later touch the formula, and so on. So basically, while handling this infant food formula, one is supposed to be extremely careful and follow the aseptic conditions so that the infants are safe and healthy. Escherichia coli. So this is the most common, everyone understands this organism. So which is basically the most studied organism in the microbiology world. Escherichia coli is a bacterium found in the digestive tract of animals and humans. Generally harmless, some E. coli are pathogenic and can contaminate food, water, and the environment. E. coli stains gram-negative because its cell wall is composed of a thin peptidoglycan layer and an outer membrane. During the staining process, E. coli picks up the color of the counter-stained safranin and stains pink. Strains that possess flagella are motile. The flagella have a peritrichous arrangement. Coming to the pathogenesis. Most E. coli strains are harmless, but some serotypes can cause serious food poisoning in humans. There are four major categories of diarrheogenic E. coli based on distinct virulence properties. So this is enterotoxicogenic E. coli, which is ETEC, enteroinvasive E. coli, which is EIEC, enterohemorrhagic E. coli, which is called EHEC, and the last one is EPEC, which is enteropathogenic E. coli. So each category or each serotype can cause the following symptoms. For example, for ETEC, the diarrhea to a severe watery stools without blood or mucus, stomach pains and vomiting are commonly seen. This is also a common cause of infantile diarrhea. In case of EIEC, invasive bacillary dysentery, fever, several abdominal pains, malaise, and often a watery diarrhea which precedes the passage of stools containing blood, mucus, and fecal leukocytes. The third one, which is EHEC or EHEC, non-bloody diarrhea through hemorrhagic colitis to the life-threatening conditions hemolytic uremic syndrome, which is HUS, and thrombocytopenic purpura, which is TTP. The last one being the EPEC. The symptoms are malaise, vomiting and diarrhea with stools containing mucus but rarely blood. Clinical features. Most varieties of E. coli are harmless or cause relative brief diarrhea. But again, a few particularly nasty strains such as E. coli 0157. Tomorrow I will be covering this organism much more in detail, which this causes severe abdominal cramps, bloody diarrhea and vomiting. Signs and symptoms typically begin 3 or 4 days after exposure to the bacteria, though one may become ill soon as one day after to more than a week later. The symptoms include diarrhea, abdominal cramping, nausea and vomiting in some people. Isolation and enumeration of E. coli and coliforms. All the methods used to detect E. coli, total coliforms or fecal coliforms are enumeration methods that are based on lactose fermentation. Most probable number method is the preferred method for isolation and enumeration of E. coli. Detection and enumeration of E. coli and coliforms can be divided into presumptive, confirmed and completed phases. In presumptive, MPN for coliforms, E. coli and fecal coliforms, the LST media for production of gas is being used, which indicates displacement of medium in fermentation vial. For the confirmed test, MPN is calculated based on the presence of gassing. You add these Durham's tubes in these tubes. So if you find a gas, then this will be used for the calculation for the MPN using an MPN table. Following the confirmed test is the completed test. To perform the completed test for E. coli, gently agitate each gassing EC tube, remove a loopful of broth and streak for isolation on EMB agar plate. Incubate for 18 to 24 hours. Look for the metallic sheen or dark centered flat with or without metallic sheen organisms. Transfer up to 5 suspicious organisms to the PCS lands and use them for the biochemical testing, which is basically the IMVIC test. So IMVIC tests are a group of individual tests used in microbiology lab testing to identify an organism in the coliform group. A coliform is a gram-negative aerobic or facultative anaerobic rod which produces gas from lactose within 48 hours. The presence of some coliforms indicate fecal contamination. I is for indole, M is for methyl red test, V is for Vaux-Prosker test, C is for citrate test. So all this in detail will be covered in my coming sessions. Association with foods, their mode of action in case of E. coli. E. coli transmission include hand washing and improved sanitation and drinking water, as transmission occurs through fecal contamination of food and water supplies. Additionally, thoroughly cooking media and avoiding consumption of raw, unpasteurized beverages such as juices and milk are other proven methods for preventing E. coli. Lastly, avoid cross-contamination of utensils and workspaces when preparing food. Fecal contamination of water supplies and contaminated food handlers, raw foods are most commonly harboring the E. coli. They are the sources for E. coli. So this will be my last slide, and coming to the references, these are the reference guides which I have gone through to make this presentation. So thank you all for giving me this opportunity to present you all about this first topic on foodborne diseases. Tomorrow, let us move on. Let us talk about the second part, wherein I will be covering more foodborne organisms and their associated topics. Goodbye and thank you.